All right, I think we're ready to begin. First, I would like to welcome everyone to today's webinar. My name is Yuan Tian. I am a scientist for content marketing at Biocytogen Boston Corporation. I will be your moderator for today's session. And I'm also joined today by two of our great panelists from the Biocytogen team. They are Dr. Kai-Qing Zhang and Dr. Sarah Chen. Both of them are from our business development team. So Kai-Qing and Sarah, would you mind saying a quick hello and telling everyone a little bit about yourselves? Maybe we can start with Kai-Qing.
Hey, yeah, thanks, Juan. Hi, everybody, and welcome to join the webinar. And I'm Kai-Qing, and just joined Biocytogen 2 years ago. And my background is PhD in biochemistry. I'm currently a BD in Biocytogen. Back to you, Yuan.
Thanks, Kai-Qing and Sarah.
Hi, I'm Sarah. I have a PhD in molecular biology and about 13 years global business development experience. I joined Biocytogen 4 months ago, and I'm very excited to be here sharing and learning about the autoimmune diseases with you guys. Thank you for joining us.
Thanks a lot. We're glad to have both of you with us today. So now I'd like to introduce our speaker for today's webinar, Dr. Frank An. So Dr. An is the Vice President of Institute of Biologics, the Antibody Discovery Division at Biocytogen. His key responsibilities include overseeing Biocytogen's internal and external collaborative antibody discovery programs, including those that have reached preclinical candidates, CMC, non-clinical safety, and IND-enabling stages across oncology, autoimmune diseases, and rare diseases. And Frank is also in charge of portfolio strategies, target inception and target product profile conceptualization, as well as project implementation and management. So Frank earned his PhD in cellular and molecular biology from Brandeis University, followed by a postdoc fellowship in immunology at the Dana-Farber Cancer Institute and Harvard Medical School. With over 20 years of experience in drug discovery across industry and academia, Frank took increasing responsibilities before joining Biocytogen, including positions at Bicycle Therapeutics, Horizon Discovery, Millennium Pharmaceuticals, which is now Takeda, and Broad Institute of Harvard and MIT. So for today's webinar, Frank will discuss the global impact of immunological and inflammatory diseases and the pressing need for better treatments. He will also introduce Biocytogen's proprietary antibody discovery platforms covered by our fully human RenMice platforms and explain how these platforms help identify monoclonal and bispecific antibodies for immunological and inflammatory diseases. You will also hear about how these platforms and techniques contribute to develop safer and more effective therapeutics. With that, before we dive into the presentation, just a quick reminder to our audience, the Q&A box is already open. So if you have any questions, feel free to drop your questions in the Q&A box during the presentation. We will do our best to address as many questions as we can after the presentation during the Q&A session. So now, without further delay, I'll hand it over to Dr. Frank An.
Okay, thanks, Yuan, for the very nice introduction, and welcome again to everyone to this webinar. So today I'm going to introduce Biocytogen's antibody discovery platforms and some applications in autoimmune diseases. Let me see if I can on the mouse. Okay, good. So today my talk is going to be in 4 parts. First, we'll do a quick review of some fundamental immunology concepts that are related to autoimmune diseases. And then I'm going to introduce Biocytogen and its antibody discovery platforms. We'll dive in some of our tools, which is a collection of a suite of fully human transgenic mice. And the first one is called the RenMap for monoclonal antibody discoveries. And we'll talk about 3 cases, TNF-SF15, or also called TL1A. And then another one is the IgE high affinity receptor, and then CD40 to illustrate how we use our platforms to make antibody discovery in the autoimmune disease area. And then I'm going to switch to bispecific antibody discovery Then introducing another platform for making bispecific antibodies, and a case study which targets OX40 pathway. Then I'm going to talk about a little bit about another very interesting modality that we just recently introduced called RenNano. And before I conclude, I'll just also mention some of our preclinical animal model capabilities of the Biocytogen. So we have a very quite sophisticated immune system. So as illustrated on the left panel here, so our immune system, the main task is to protect, our body from disease-causing pathogens, and so that's the role of our immune system. And it has an army of immune cells to carry out these functions. In the interest of time, I'm not going to delve into details, but just to point out that here we have T cells and B cells that belongs to the adaptive immunity part. And also we have dendritic cells or DCs, macrophages, and the mast cells, granulocytes in the innate immunity category. So our immune system has to work well. And one of the fundamental and actually quite amazing things is that it has to balance the task of killing the foreign antigens, pathogens, but also not doing any harm to our own body. So we know there are two types of immune responses. One is called the type 1 that is responsible for killing the invading pathogens. Another is called type 2 that more involved in regulations of the immunity, etc. So these two types, both types of immune responses needs to work, okay, in the balanced way. Balanced means it cannot, it has to it cannot mistake our own body cells, organs, tissues as foreign, so it won't attack those organs. And the second is that when the pathogens are gone, it has, when they mounted an immune response, but when antigens are gone, they need to find a way to tone down the immune responses. So either if it loses capability, then it would cause autoimmune diseases or fibrosis and other problems. So the situation is very much like in the Goldilocks story. So everything has to be perfect. It cannot be too hot. It cannot be too cold. But we are living in the real world, so it's not perfect. So our immune system can break down, and the tolerance can break down. And when that happens, our immune system will attack, the healthy organs by mistake. So that happens, and when that happens, we have an autoimmune disorder. So there are over 100 autoimmune diseases. So this picture actually shows a lot of the immune diseases organized by different organs, and all the organs in our body can be impacted by some of the autoimmune diseases. And it is estimated that 10% of the global population has had autoimmune diseases. And in the US, there are 50 million patients. And this disease tended to cluster. That means a patient can tend to have more than one autoimmune diseases. And for some reason, that 80% of the autoimmune disease patients are women. On the therapeutic side, and it's interesting that other than the small molecules, okay, the antibodies is the #1 modality, okay, that people have been working on trying to come up with autoimmune disease drugs. So with this introduction, let's turn to the antibody discovery part of the talk, and especially that how at the Biocytogen, we use the platforms, our own proprietary platforms, to do antibody discovery. So first is a brief overview of Biocytogen. So Biocytogen is a global biotech company with over 1,000 employees, headquartered in Beijing with a huge, large, very, very large animal facility in Jiangsu province. And we have about 100 people strong team in Boston, United States, and a business office in San Francisco on the West Coast, and another business office in Heidelberg, Germany. So we brand ourselves in two parts. So the first part we call the Biologics, which is engaged in antibody therapeutics discovery, research and development, and out licensing. And another arm is called BioMice, that is involved in CRO preclinical products and services such as gene editing, pharmacology services, animal models, humanized mice, etc. These are the modalities we have been working on antibody discovery. So the first one is a monoclonal antibody platform. And we also have a bispecific antibody platform, and derived from the bispecific antibody platform is the bispecific ADC platform. And we also have a nanobody platform, and a recent member to this collection is fully human transgenic TCR platform. And also we have some difficult to target classes approaches like the TCR mimic platform and the GPCR platform. And all these modalities are predicated on a suite of fully human transgenic mice, collectively called RenMice. So, so these are the overall the antibody discovery platforms we use at Biocytogen. So specifically, first let's talk about the first member of this collection, which is called the RenMab. Ren in Chinese means human. Mab stands for monoclonal antibodies. So in this mouse, okay, the heavy chain was the mouse heavy chain in this mouse was totally replaced, entirely replaced by the human heavy chain, those right bars, VDJ sequence. So in this mouse, the entire heavy chain, it carries the entire human heavy chain repertoire. On the light chain side, the mouse kappa light chain was also totally replaced by the human kappa light chain, the right bars. So in this mouse, RenMab, it carries the entire human heavy chain repertoire and also the entire human kappa light chain repertoire. So that's as diverse fully human transgenic as you can get. Here's an application with the RenMab and also some other RenMice strains. It's something we engaged about 3 years ago. It's called the Project Integrin, which essentially is a large-scale early antibody binder discovery for over 1,000 targets. So we were motivated by the gap between the number of targets that actually through which there are approved drugs, and also the potential antibody druggable targets. So its difference is about 1,400 and about 100. So we wanted to fill this gap with our antibody binders and the preclinical candidates to aid the discovery efforts of the entire community. So during the last 3 years, this is what we achieved. We have been working on over 900 projects, and we created about 500,000 sequences with characterized binders. Characterized means we have the binding data on them, the affinity, cross-reactivity. If there are tumor-associated antigens, we even have the internalization data. And we also generated over 40 preclinical candidates. So these are the mid-stage molecules, that's ready for CMC. And these are the targets, covers almost all the disease areas. And we have been very active in forming partnerships to make full to make full use of those molecules. So here gives you a flavor of the targets we have been working on under Project Integrum. So this is organized by disease areas: oncology, inflammation and autoimmunity, neuroscience, metabolic and cardiovascular diseases, just to get a flavor of what we have been working on. Okay, now I'm going to talk about three applications, monoclonal antibody discovery cases using the RenMap. So the three cases targeting three different areas. First is the inflammatory bowel disease, or IBD, the allergy and anaphylaxis, and then Sjogren's syndrome. So the first is IBD. So IBD is actually composed of two distinct diseases, ulcerative colitis and Crohn's disease. So these are two different diseases, although they share some features and symptoms like diarrhea, abdominal pain, bleeding, weight loss, etc. They also carry cancer risks. So worldwide, there are almost 5 million cases of IBD, and the US is particularly high in both prevalence and also incidence rate. And it has a complex pathogenesis, but generally the consensus thinks that it is the Th17 and the Th1 driven diseases, as illustrated on this panel here, on this picture here. And since 1998, so the treatment or management of IBD, we sometimes call that advanced therapies, which includes biologics, JAK inhibitors, and this S1PR modulators. So they have made a tremendous impact on the management of IBD. So the first TNF, in terms of antibody drugs for IBD, the TNF alpha blockers, the first approved was infliximab in 1998. Then integrin blockers, vedolizumab, approved in 2014, that targets integrin alpha-4 beta-7. And then up to very recently, risankizumab, that was approved for, actually this year, for UC, but has the best-in-class potential. However, despite these successes in IBD management, there's still a very urgent unmet medical needs, because there are still a significant percentage of patients failed to respond. They can be categorized into primary and secondary non-responders. And also in terms of needs for surgery for these patients, that rate, sometimes the rate is still very high, particularly in CD, where it's estimated that 50 to 80% of patients would need surgery because the therapeutics could not work on them, so they needed to do surgery. So we very urgently need alternative targets and orthogonal MOAs with increased efficacy and long-term disease control, and also more convenient administrations by the patients. So here came the next rising star, targeting TNF superfamily member 15, or it's also called TNF-like ligand 1A, or TR1A. Okay, so it's very busy. Well, busy is not the right word. It's a very popular field, and a lot of companies are chasing a few molecules. And you can see here, three multi-billion-dollar deals happened in succession in last year by Merck, Sanofi, and Roche. So what's this molecule? So TNF-SF15 is a by definition, but you can tell from its name, it's a TNF family member, and is expressed by endothelial cells constitutively, but is also can be expressed by DCs and macrophages. It's upregulated in IBD. And what's interesting about its function is that it sits on top of a lot of inflammation pathways, okay, and regulate their activity and promotes their activities. And also they promote fibrosis. So so that's why you know, it acts like a master regulator of this many inflammation pathways, and it's clinically validated targets. This is the, you know, almost $11 billion molecule Merck bought. It's called the PRA23, and it showed actually very promising phase 2 activity for both UC and the CD, and here is the phase 2 activity for UC. So very good best-in-class potential for this molecule. So we also, you know, worked on this target, and here is generic our antibody discovery workflow, that we would start with immunization of RenMice, and we sometimes use KO. KO means knockout strain, and so that's first step is immunization. Now we have a beacon, which is a single B cell selection instrument that can sort thousands of B cell clones in a day, and then we'll check the binding activities of those clones, and we'll express the protein, and then doing further characterizations in terms of cellular binding, you know, affinity measurement, and you know, blockade between receptors and the ligand, and internalizations, etc. So we followed this. Oh, here is just one point I wanted to make, is that why we use KO mice, okay, RenMab mice on the knockout the target gene, the target mouse copy on the RenMab background. This is because we want to reveal additional epitopes that are, you know, otherwise would be hidden because they are identical between human and mouse. Okay, so by knocking out, removing the mouse copy, these epitopes would be available for immunogenicity. So here is just to give you one example of what we got obtained from our efforts targeting TL1A. So here is our 3 clones, A, B, and C, and you can see that you know, they bind really well. This is SP. This is a BLI, and so you can see that they bind really well. You know, almost, you know, a flat line in a dissociation phase, and relative either equivalent or better than the 2 benchmark molecules. And here is the reporter functional blockade data, and you can see that at least 2, the blue and the red here, were more potent than the benchmark molecules, which, you know, their curves here was more. So the 2 clones, A and B, were more potent than the benchmark molecules in this reporter assay. Okay, so that was about TL1A. The second case is about we're also interested in allergy and anaphylaxis. So as we all, some of us has pretty good personal experience about what allergy means to us, and essentially is when the immune system overreacts to something that in the environment that is actually normally innocuous. So that's not harmless. Okay, so there's a lot of these things. It's like pollen from plants. This is actually electronic scanning image of pollen from sunflowers. And anaphylaxis is a very severe form of allergy, and sometimes could be fatal if not treated. So allergy, there are two key players mediating the triggering of allergy reactions. They are IgE, and IgE and its receptors. So there are two receptors for IgE. One is a high affinity receptor called Fce epsilon receptor 1, okay, or FCER1, that's how I would call it. And another is a low affinity receptor called CD23. And they bind in a very stable complex, and FCER1 would transmit a signal to the effector cells, and the CD23 is responsible for antigen presentation, for IgE-mediated antigen presentation, and also B cell production of IgEs. The pathogenesis mechanism is divided into two stages. First is called IgE priming. So IgE essentially the allergens would increase the production of IgEs, and then the second process is triggered. Then IgEs would cover the effector cells that includes mast cells and basophils. And then the second step is by additional, allergens would bind the allergen-specific IgEs and cross-link them, and that would trigger signal transduction and activation of those effector cells, and they release the effector molecules like histamine that's regarded as allergy symptoms. And this is an area that has very intense research on antibody drugs. And one of the very successful ones was omalizumab. And it has, last year, it sold for almost $4 billion. And it has only 4 indications marked by the green, and a number of related possible indications as well. So it's a good market and interesting pathways to work on. So here is our what we did, and here is the the workflow for antibody discovery. Now here we use the RenMap knocked out mice, and we're working on. We are focused on the receptor side rather than the target. The antibody side, the IgE antibody. The omalizumab is actually a IgE neutralizing antibody, but we're interested in team at Biocytogen is interested in anti-FCER1A, which encodes the alpha subunit or the binding moiety for IgE. So here is, it's like we immunized 20 mice. We originally scored 80 positive clones, and then we narrowed it down to a smaller number. Now, additional characterizations are ongoing. So here I just wanted to show you the affinities of these some of the clones we obtained. Here's binding. Here's the Y should be the KD values here, and here's for human. That is for monkey FCER1A. So I forgot whether I explained that some of the data details were redacted because, as you can see here, because of legal recommendations. Okay, so then we switch to the third case, which targets CD40. Okay, and for many of us who learned immunology in college, that we know that the CD40 and the CD40 ligand, they play indispensable and critical roles in germinal center formation and also in class switching, so in making antibodies. So this is a very fundamental target in immunology. And that is, what I just described its roles were well established in mice. Here's the knockout, CD40 knockout mice, and you can see it has a follicle here, but compared to wild type mice, it doesn't have this middle thing, okay, which is the germinal center, important for antibody maturation. And also, it can produce IgM, which does not require class switching, but anything after IgM that requires class switching in the CD40 knockout mice, there's none. So here is, for example, IgG1 in non-immunized mice here. The black dots means the wild type, and the open circles are the knockout. So you can see in the knockout mice, CD40 knockout mice, there was no IgG1 in the resting naive mice. Even that's the same is true if you immunize the mice with an antigen, then still there is no IgG1 production in these knockout mice. And very interestingly, that all this people are observed in mice were replicated in humans. Okay, the human CD40, either the ligand or CD40 mutant individuals will not have class switching event. In other words, they don't produce IgGs or IgAs, etc. But they produce high level of IgMs. So that is very interesting. And then also it's clinically validated. Novartis has this anti-40 antagonist, iscalimab, and it showed positive signal in the Sjogren's syndrome in their phase 2 trial. So this is just a fundamental molecule involved in immunology. So we have a molecule that has reached the preclinical candidate stage, and here's some of its data. And here is the binding by SPR, and we have our PCC molecule here, human and monkey CD40 binding, and we have two control molecules that have been used in clinics. So just look at this table here, look at the last column, that's the KD values, and our PCCs were either equivalent or better in terms of affinity to these benchmark controls. And here is the additional functional data. Here's the reporter blockade data. So this is a CD40 ligand-mediated signaling events, and these anti-CD40 antagonist antibodies can inhibit this signal. So, and our molecule is this PCC, which is this blue triangle line here, which is most potent relative to the other three benchmark molecules. We sometimes also call them positive controls. So that's what the PC stands for. So overall, that these data indicated that in vitro, our PCC molecule was more potent than the benchmark antibodies. And then we put this molecule to test in vivo, and here we use a pharmacodynamic model, or PD model. It is the immunization of mice with overwhelming or OVA, and then we give the mice antibodies, and because it blocks the class switching and IgG production, we can measure the titers, the IgG titers against OVA. So here is our molecule, and here's the benchmark, one of the benchmark controls. And here, we dose the animal at the 4 different doses, 4, 2, and 1. And here we observe the titers on days 10, 17, and 21. So first you can see that, we see a dose-dependent inhibitory effect of OVA titers. Here, the higher dose, the animals tend to generate less antibodies, here's the Y scale, the log scale here. And so it's yes, so it's a dose-dependent inhibition of OVA titer. Now, if you compare our molecule versus this benchmark control, and overall, you can feel that ours was more potent. For example, if you look at 1 mg per kg dose on day 10, all the mice treated with PCC were here at a much lower level than the mice treated with the blastelimab, which is a benchmark and NF-CD40 antibody. So that's provides us with in vivo evidence that our molecule also outperform the known anti-CD40 clinical molecules. Okay, so that's the all the cases were about monoclonal antibodies, but also as we know that which monoclonal antibodies are the classical modality in autoimmune disease therapeutics. But now we have bispecific antibodies, and it has become popular in treating autoimmune diseases. In the first half of the year, J&J spent almost $2 billion to purchase 2 molecules, which are bispecific molecules involved in treating atopic dermatitis. So we also, have a platform we call the RenLight. The light stands for Common Light Chain Technology. So this is how the mouse was constructed. So the heavy chain side that carries entire human heavy chain repertoire, as I described for the RenMab strain, and also here but on the light chain side, there's only one human light chain gene that replaced the entire mouse kappa chain locus. So the reason was that we liked this platform for bispecific is that it solved a very troublesome problem, which is the mispairing of the light chain and heavy chain. As illustrated here, with the common light chain technology here, you can express 3 molecules, the light chain and 2 different heavy chains that targets 2 different targets. And then there's a very good chance, over 90% of chance, that you can get the functional, readily assembled bispecific molecules here. However, if you do not have the common light chain, you have 2 different heavy chains and 2 different light chains. Although the heavy chains can be assembled correctly through knob and hole technology, but there's a mismatch of the light chains to the assembled heavy chains, then only a fraction of your antibodies would be the desired bispecific molecules. Okay, so let's see one case where we generated a biparatopic anti-OX40 molecule for treating atopic dermatitis and probably other type 2 diseases. So brief intro about this molecule. OX40 is expressed on T cells and is a co-stimulatory receptor and plays very important roles in T cell, a lot of T cell functions here. But what's very interesting that attracted our attention was that its role in atopic dermatitis pathogenesis, because it sits upstream, just like TL1A does. It sits upstream of all these very important TH pathways that regulates the atopic dermatitis pathogenesis. So what is our goal to work on this? So the field, the OX40 and OX40 axis, here are some of the clinical molecules that targets OX40 and OX40 ligand. And also, it's not shown here is 800-pound gorilla in the room, which is dupilumab, that also works, very well in atopic dermatitis. So here, our team tallied up the anti-OX40 benchmark molecules, rocatinlimab, or this Amgen's molecule, also called the KHK4083, And in blue here, that's a anti-OX40 molecule. And also the dupilumab in red. And here's in green is the anti-OX40 molecule from Sanofi. And here is another anti-OX40. The purple is another anti-OX40 molecule in clinic. So when we look at the different readout, clinical readout such as EASI-75, EASI-90, and IGA 0/1, you can see that the Amgen's molecule, the rocatinlimab, actually worked, either equivalent or slightly better than dupilumab, which is the best-selling AD drug out there. So in order for any newcomers to have a commercial success, okay, and it has to be more efficacious and more convenient to administer and have a manageable safety profile in order to unseat dupilumab as the top drug in AD. So the goal is for us to come up with an anti-OX40 molecule that is better than the existing clinical molecule, and so that by inference, hopefully that our molecule would be better than dupilumab. This is the molecule we constructed called the BCG28, and it's a common light chain bispecific targeting that targets 2 epitopes on OX40. Okay. And here is the affinity information that you can see the biparatopic antibodies has improved affinity for the targets relative to its individual arm, and also better than the 2 benchmark molecules. Here are some of the in vitro assays we performed. Here, in blue is BCG28. That's our molecule, the biparatopic OX40 anti-OX40 molecule that shows equivalent activity to rocatinlimab, which is the red line here. And similarly, when we measured the cytokine IL-2 secretion in PBMCs, they also demonstrate similar activity. And here, when we tested in an assay that more closely resembles the in vivo stimulation of T cells, and you can see the differentials here. Our molecule, which is the BCG28, the blue line, outperformed the benchmark, the rocatinlimab, on the red line. So in this assay, that more resembles the in vivo T cell activation. Here's the in vivo data. The model is GVHD, and our molecule protects the animals from death caused by GVHD reactions much better than rocatinlimab and all the other anti-OX40 benchmark controls. So this is in the wild type IgG1 format, and I'm not going to show data in the interest of time, but the drug forms of our molecule also outperformed these 2 benchmarks in their respective drug forms in this GVHD model. Here are some of the more assays regarding the safety aspects of this molecule. Here is the agonism, which we do not want. So here's the reported assay. So the benchmark control has much stronger agonism than our molecule. Here is the agonism in primary T cells. Then again, the benchmark had a stronger agonism, which was not desired than our molecule. And our molecule, even at very high concentrations, would not activate PBMCs to release IL-2, which is just good. And also our molecule, which is this blue bar here, that has very little immunogenicity inferred by this THP1 internalization assay. Here are some also other properties of our lead molecule BCG28, which is biparatopic OX40 molecule. First, the solubility on this PEG assay. It demonstrated very good solubility, so we estimate that it can make a high concentration solution suitable for subacute dosing. And here's the PK data. And we introduced the half-life extension mutations into the IFC part of BCG28, so it doubles the half-life of the benchmark controls. Here's the developability data, and say 14-day, the molecule was subjected to 40 degrees incubation for 14 days, and we measured by different analytical methods that the molecules remained intact. And also here's a binding assay. You can see before and after, the two curves essentially overlapped. So that indicates very good stability. So overall, when we tallied up all the activities, properties against these two benchmarks, and anything better than the KHK 4083 rocatinlimab, we give it a letter A. If it's the same, we give a letter B. If it's inferior, we give a letter C. So you can see that the BCG 028 is the top molecule. Hopefully we hope that it will translate in clinic. Here are some additional targets that I did not have a chance to talk about in the immunology and the autoimmune disease area. And here, just in the last few minutes, just mention a couple of other things. Here is the third member of our RenMice collection, which is called RenNano. And we heard nanobodies from camelids. So these are the smallest building blocks, single domain antibodies or heavy chain only antibodies. And we generated this RenNano mice. And so this is fully human transgenic mice that only produce this single domain antibodies. You can use the single domain antibodies with IFC or without. So we started this year a program called the Nano 100 initiative that we're trying to discover these nano antibodies against 100 also high-value targets. And some people might have seen this beads on a string type of structure. These are the multi-specific antibodies generated by people using the camelids VHH single domain antibodies. And hopefully in the near future that we're going to replace these VHH camelids antibodies with the fully human RenNano antibodies, because that would save a lot of time in humanization of the camelids antibodies. Okay, so very quickly in the last 2 or 3 minutes to say a few words about our autoimmune preclinical models, because they are also a very important part when you study autoimmune diseases, because what I did was all about antibody discovery, but without good in vivo models, you cannot characterize your clones that well. So here is the overview of our animal models, mouse models in inflammatory and autoimmune and metabolic diseases. Not going through details, but they're covered, various organ systems, skin, respiratory, CNS systems, and altogether, it lists over close to 60 preclinical models. This slide shows you 2 examples. One is the 2 examples. One on the left is the collagen-induced arthritis model called the CIA. So we validated this model with anti-TNF alpha antibody, this red line here, and also supported by our histology data. On the right is an IBD model. It's called a DSS-induced acute colitis. And here, we generated the model, and also we used, this is a antibody against the CXCR2. These 2 lines, the purple and the orange lines, you can see that these mice that received anti-CXCR2, here their body weight loss was improved relative to the controls. Another 2 models here. Here is the asthma model triggered by the intranasal administration of human TSLP and overwhelming. And you can see here that here we used the benchmark control antibody, TSLP antibody from AstraZeneca and Amgen, abbreviated as TZP. Here you can see it reduces in the last two groups, reduces infiltration of eosinophils into the lung. And here's a TARC, reduce the level of TARC, which is a chemokine produced by the lung cells, and also the OVA specific IgE levels in the blood. This is the last model is about pre-stain induced lupus model. And you can see here validated with anti-CD40 antibody, which is the green bar versus the controls. Okay, so well, before I conclude, I'd just like to give a big thanks to our project teams that provided the data that I just spoke to you. And also thanks for our marketing team for organizing this webinar. And with that, thank you very much for your attention and for coming to my webinar. And if there's still time, I will be happy to answer any questions. Thank you very much.
Thank you, Frank, for the great presentation. I think we've already received a few questions in the Q&A box. So let's get to a couple of them. And to our audience, if you haven't submitted your questions yet, feel free to post them there now. Okay, so the first question here is, can you elaborate on the unique features of the RenMice platform that distinguish it from other transgenic mouse models used in antibody discovery?
Okay, that's a very good question, and I'll try to answer that. But I'm more on the antibody discovery side, and these mice were generated by our gene editing group. So I'll try to answer, and but if you have any remaining questions, feel free to ask, and we'll get the answers to you. So anyhow, I think ours, first of all, is that it carries the most diverse human immunoglobulin heavy chain, and in the RenMap mouse, also the kappa light chain repertoires, and a lot of other fully human transgenic systems out there. Some of them just used part of the heavy chain repertoires to construct their mouse strain. And clearly there are successes. A lot of the antibodies that are drugs, antibody drugs, or from those systems. But just in terms of the repertoire composition, I think ours was the most diverse. And the Regeneron's Veloci mice were constructed similarly, but we use different technology. It's a homologous recombination with very large piece of genomic DNA's replacement. So these are, I think, the more fundamental distinctions. And also since we use in situ to replace the mouse locus, so it carries as many it kept intact as many regulatory elements on the genomic DNA. And some other companies use the transgenic approach to introduce the human genes, and so the regulatory aspect might be different. These kind of I think the most important aspects, but there's a lot of technical differences between different systems. So if you have any further questions, I'll ask the experts in our gene editing group and to get an answer for you. Thank you.
Thanks, Frank. And another question we received is, are disease models made in RenMice background?
Okay, RenMice are specific. When we say RenMice, it means RenMap, RenLight, and RenNano, and RenTCR. So these are used for antibody and TCR discoveries. So that's we call them fully human transgenic mouse strains. So this is different from the humanized mouse that I talked about. That they means that we introduce the human target, like human CD40 or human uh, TNF and the TNF receptor into the mice. Because actually, that's a very good point. I wanted to make that point, but I forgot. So, in particularly immunology, okay, because the anti-human antibodies, okay, do not recognize the mouse counterpart. So we have to use these humanized mice where we introduce the human target instead of using the mouse counterpart. So that's a very, very critical step particularly in immunology when you have, like a cytokine or receptor has different subunits, and that can be very cumbersome to generate a system where you can study anti-human targets. But at Biocytogen, it's one of our strengths. We have generated hundreds of these humanized mouse strains for the community to use. Thank you. That was a very good question.
Thanks, Frank. Just to add on that, most of our disease models most likely made in humanized mice or white mice background. So that's another, advantages.
Yeah. Yes.
Okay.
Thank you. Yeah, we have the B6 background and also biopsy background.
Yes. All right. Next question. What is your general procedure to construct bispecific from single binders?
Okay, that's a very good question. It depends really on your purpose, because clearly, when you have a bispecific, each arm would target, either two different epitopes or two different targets. So it's really depends on the biology, what you want. Okay, out of those bispecifics. Usually you want more, okay, than targeting those two targets or epitopes separately. Like in the case of anti-OX40 by paratopic case, we chose the two arms. One arm would block the interactions between the OX40 ligand and OX40, and another arm would help stabilize that molecule on the target so that we can have a longer residence time, longer inhibition. For bispecific ADC, we wanted something else. We wanted the two arms would synergistically would help each other in facilitating the internalization of our antibodies that carries cytotoxic payload, so that in this way it would kill the tumor cells more effectively and also can generate certain added safety because you can choose the arm so that it does not get into the cells if it only express one antigen, which a lot of normal cells might express one antigen, but the tumor might carry both antigens. So essentially you added the binding specificity. Okay, so you after you know your purpose, then you try to look for antibodies clones that would give you those properties. Of course, what we usually do is that we have 5 good clones target one target, and 5 good clones that target another target, 3 or 5 or 7. Then you do a mix and match combination. So then you screen. There's no other way. You have to screen based on the criteria you set. Okay, so that's how we generate a bispecific molecule, generally speaking.
Thanks, Frank, for the detailed answer. I guess we'll just handle one more question due to the time. So what will you do if knocking out the gene leads to defects in immune system development?
Ah, then then you're out of luck. Okay, it's just that just means you cannot do that, because once suppose that you want to make CD3 and anti-CD3 antibodies, when you knock CD3 out, then the mice would not mount any immune reactions. So in that scenario, you have to work some other ways. So when I say when we use knockout mice, usually that refers to something that will not interfere with the immune responses. That's a good point.
All right, thank you, Frank. Unfortunately, I don't think we have time for any more questions, but feel free to contact us through our website. For further questions, we will be happy to get back to you. With that, again, a big thank you to Frank and our panelists for sharing their insights, and of course, to everyone who joined us today. Just a quick reminder, if you missed anything during today's session, this webinar will be available to watch on demand. You can request it or find it on Biocytogen's website or our YouTube channel. And that said, feel free to explore our website and YouTube channel to learn more about our projects and assets. You can also search for RenMice platforms on our website to see a broader list of targets and the current progress of discovery. And again, if you have any further questions about the webinar, about RenMice or Immunologics, or anything else we offer on the website, feel free to contact us through our website, or you can directly contact our BD licensing team for the Immunologics at the bd-licensing at biocytogen.com. All right, with that, I think that's all we have for today. Again, thanks everyone for joining us. As the holiday season is almost here, we hope you have a warm and joyful winter break, and we hope to see you next time. Thank you.